So I'd like to start by thanking Mills College for the great honor of being invited to be here. And I'd like to especially thank Chris Russell for being here. She has been involved in biology and some of the policy issues around it from the very earliest days of the molecular biology revolution. And it's an honor to spend time with her and with you while I'm here. Now, your motto here is there are different paths to one destination. And my talk today will be about my path to a destination for leadership in science and the work that I've been trying to do at different stages in my career and how I've learned from that how to think about perhaps ways that we can make science better. And my story starts in graduate school at MIT, where I was at the same time as Lisa Ori, and where I studied cancer. And I just want to talk about what I learned there. So I, as a graduate student, studied this very fine organism, which is a rat. And this particular kind of rat got a particular kind of cancer. It was a brain cancer, a neuroblastoma. And we were trying to understand the genetic basis of this neuroblastoma. And I was working in a lab where everyone else was studying human cancer, Bob Weinberg's lab. And there were many reasons that it seemed much more interesting to study human cancer, and it seemed like kind of a weird thing to be studying this rat. But the reason that this rat was interesting was that the cancer that arose in this rat was associated with something called a tumor antigen that could allow the immune system to reject that cancer and allow the animal to survive. And this tumor antigen we characterized in collaboration with someone who had very different expertise from us, a man named Mark Green, who was an immunologist. And ultimately, my little piece of the story is that I identified the gene, I cloned the gene that gave rise to this tumor in the rat, and determined that this gene actually encoded a protein that sat on the surface of the cancer cells. And that was why those cancers, so first of all, that was the tumor antigen, and that was why the antibodies could attack the cancer, because the cancer couldn't get rid of that gene without stopping being a cancer. And so it maintained that, and it's sort of the same thing that made the cancer grow was also its greatest vulnerability. Now, this was an interesting insight, but it seemed very rat-like, and there were lots of reasons for thinking it wasn't relevant to human neuroblastoma. But after my work was finished, and at that point it would have been stopped, other scientists picked up the thread. So Mark Green developed really good reagents that allowed you to detect this molecule, the tumor antigen. And then working together with a medical scientist at UCLA named Dennis Slayman, they looked at a lot of different human cancers, and they discovered that, sure enough, it's not altered in neuroblastoma, but it's altered in a subset of breast cancers. And in fact, at the time that that was discovered, that subset of breast cancers was the most lethal and most dangerous form of breast cancer for women to have. The gene involved, we called NU for neuroblastoma, but it's also called HER2. And after that discovery was made, so there was a relationship to human cancer, but it took this medical group and the immunologist to find it. And after that discovery was made, the story migrated to one of the brand new biotech companies that was growing up in the Bay Area called Genentech. And Genentech made an antibody against the human version that could be tolerated by humans. And that antibody is called Herceptin. And it's now used to treat about 100,000 women a year with breast cancer. And through that antibody, now that particular form of cancer is the kind you actually want to have because it is one of the most treatable and one of the most curable. So what I learned, I learned a set of things from this, right? And what I learned is that scientists build on each other's work and that it takes a lot of different people with different expertise to make progress. And I do not want to exaggerate my contribution to this story. I'm one of hundreds of people, maybe thousands of people who contributed to this work. But I did learn from this that science works, and if you really try to deeply understand something, and if you work with other people with different expertise, you can make progress even against some of the most terrible diseases of people. And that is an inspiration for me, and I think an inspiration for all of us in science to realize that even when you're doing something that seems awfully obscure, and even when you're laboring away and the experiment hasn't worked 10 times, maybe that 11th time you'll do something that someone else can build on, and that ultimately will lead to progress that makes a difference.